Hello and welcome to the webinar. I'm Melissa Romero, Forensic Science Consultant and CNN Media Group, and I will be moderating today's event. This webinar is entitled Accelerating Drug Discovery, Enhanced Chemistry Strategies for Novel Chemistry Entities, ADCs, and Emerging Modalities, and is sponsored by Eurofins Discovery. CNN works with sponsors to identify topics of interest and value to CNN's audience and consistent with CNN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. A few technical items before we begin. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast at any time, please log in again according to the instructions you received earlier. You are also encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNN does not endorse any company products or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. The presentation today is being sponsored by Eurofins Discovery. The company has built its industry-leading reputation on successfully delivering best-in-class drug discovery services to any organization, large or small. As a global leader in drug discovery for more than 40 years, Eurofins has strategically amassed a deep and broad range of over 12,000 assays for every type of research, as well as a full team of industry experts across disciplines to collaborate with you. No matter where you are in the preclinical process, or if you need full end-to-end solutions, its experts support you every step of the way with purpose, precision, and innovation. During the presentation today, we will hear from two speakers. First is Matthew Horowitz, Project Manager and Head of Scale-Up Laboratory at Eurofins Discovery. Matthew earned his PhD at the University of North Carolina, focusing on asymmetric catalysts and total synthesis of steroidal alkaloids. He completed postdoctoral fellowships focused on photochemistry and fluorine chemistry, and subsequently worked in discovery process chemistry and process R&D at Johnson & Johnson. Currently, he supports teams in discovery chemistry at Eurofins Discovery and is based in Murcia, Spain. Our second speaker will be Sujay Basu, Director, Discovery Chemistry at Eurofins Discovery, located in India. With over 24 plus years of experience in medicinal chemistry and drug discovery, Sujay has led the multidisciplinary chemistry team since 2006. He holds a PhD in medicinal chemistry from Andrew University and has successfully delivered 4 preclinical development candidates and 2 clinical candidates. Sujay is the co-inventor of Lipiglin, India's first approved drug for dyslipidemia, type 2 diabetes, and NASH. And I'll now hand things over to Matthew.
All right, thank you very much for the kind introduction, and to you out in the audience as well for tuning in today. We'll go ahead and get started. I'm Matt Horowitz, and I'm here today with my colleague, Sujay Basu, and today's webinar is all about figuring out how to take the right decisions and move faster when it comes to drug discovery programs. We'll talk about small molecules, which have formed the basis of a lot of quote-unquote traditional drug discovery programs, but we'll also talk about more recent developments in targeted protein degradation and antibody-drug conjugates, or ADCs. Just to briefly introduce ourselves before we get into the talk, Sujay is a director in medicinal chemistry and brings a wealth of experience in structure-based drug design, SAR for lead optimization, and generation of preclinical candidates. He's based in our site in India. And I'm Matt, a project manager in our site in Spain, driving drug discovery programs with innovation in chemistry and applications in new modalities. With our teams in Europe and Asia, we help our clients in pharma, agro, and biotech to advance their programs as quickly as possible by taking the right decisions. At Eurofins Discovery, our vision as a global chemistry service provider is to be able to provide a full range of services, from synthetic chemistry, the preparation of new compounds, to medchem and compchem, or the design of new potential medicines. But beyond a solid base, we also have a competitive edge in that we are fully leveraging robotics and automation, both for synthesis and purification, in order to generate new compound libraries and explore SAR vectors faster than ever before. We're also able to follow through with downstream CMC support, including the production of preclinical tox batches, as well as GMP batches and drug product development. Our chemistry support can either be accessed as a standalone service or as part of a fully integrated drug discovery collaboration, where our chemistry team works in close collaboration with hit-finding teams, as well as biochemists and biologists who execute downstream assay cascades that are designed and orchestrated by dedicated program managers in collaboration with scientific project leads. Long story short, if you want to find a new medicine, we have the firepower to get you from start to finish and provide you with everything you need along the way. Let's be honest, drug discovery is a challenging task, no doubt about it. Getting from each step in the process to the next involves very careful planning and decision making in order to make sure that all the relevant factors in the specific program are being addressed and that risks and liabilities associated with the candidates are being minimized. Everyone in the industry knows that this is a competitive field and that many therapeutic areas have a crowded IP landscape. In today's talk, we want to address situations like these and answer the question, how can I identify novel starting points for a drug discovery program? Once we've found a starting point and we want to get answers to the most important questions as quickly as possible, it's important to leverage automation as much as possible for the exploration of efficacy trends and structure property relationships. This applies not only to small molecule-centered programs, but also to programs focused on targeted protein degradation and antibody-drug conjugates, where it's important to find a way to manipulate highly cytotoxic materials in an efficient way. In short, drug discovery requires experience and innovation, both in terms of scaffold design as well as implementation of technologies that accelerate SAR interrogation. Advancing a program in drug discovery requires being able to efficiently go around the design, make, test, analyze cycle. We start each cycle with a testable design hypothesis, which might be based on some combination of data coming from biology, medchem, compchem, AI, searching of patents and academic literature, and just intuition. And we then try to evaluate that hypothesis using chemical synthesis and a program-specific assay cascade designed to answer the most important questions first and minimize risks. To be able to move through these cycles efficiently, it's possible to use automated synthesis systems like our ChemSpeed robot that we can see here on the right, which we use for the production of targeted libraries. When it comes to cytotoxic payloads for ADC applications or thalidomide derivatives for targeted protein degradation, this is also where we implement high containment protocols in our dedicated labs to support these activities. Once we've analyzed the properties of the molecules we've made, which might include potency, ADME properties, and off-target activity, we form a conclusion that allows us to restart the cycle with an improved perspective and a refined focus. And as we go around the cycle, each iteration brings us closer to a candidate that can be advanced into clinical trials. But to get there, we have to make the right decisions, which means we have to have all the right information as quickly as possible. And that's really the key focus of what we're going to be talking about today. How to identify the right solutions for your drug discovery program and accelerate at every stage. We'll talk about what the specific enabling technologies are that you can use in order to drive your programs in the right direction and reach each milestone faster. We'll then go through a recent case study in drug discovery with my colleague Sujay to explore how it's possible to innovate in a crowded IP space to arrive at novel, potent, patentable compounds. And we'll also take a look at drug discovery in the context of targeted protein degradation as well as antibody-drug conjugates. Finally, we'll end by talking about how chemistry collaborations work at Eurofins Discovery. Let's start by talking a little bit more about enabling technologies. Enabling technologies give us the opportunity to accelerate and get more data at every stage. Docking, for example, allows us to optimize the interactions that our compound has with a biological target of interest through detailed computational studies, allowing our design process to be more rational. We also have the ability to screen compounds virtually in a high-throughput way and to use AI to guide our design process. With our recently developed AI tool, Sapphire, we've made a very user-friendly interface to be able to quickly get insights about the predicted ADMET properties of novel molecules using machine learning models built on large, robust internal datasets. Targeted libraries have also become increasingly important for searching new chemical space quickly in order to do hit expansion and move from a hit to a lead. In order to generate these libraries, we use the type of automated robotic synthesis platform we saw in passing earlier. The same robotic platform could also be useful for synthesizing hits coming from virtual screening, for example. However, it often happens that medicinal chemistry campaigns hit a roadblock in the form of a tricky reaction or a difficult step in the synthesis. And for those cases, we apply high-throughput experimentation, or HTE, to make sure that we're able to find good conditions quickly. To understand the physical chemical properties of the targets being generated in medicinal chemistry campaigns, it's important to have a reliable partner for ADME as well. Minimizing the synthesis to ADME turnaround time is really critical for being able to understand the structure-property relationships in a detailed and efficient way. Furthermore, as you start to find targets of significant interest, it's often useful to identify a solution for high containment work, as well as scale-up operations. In our chemistry labs in Spain and India, we've made significant investments in order to co-locate the enabling technologies that accelerate our clients' drug discovery programs and minimize turnaround times. So, how exactly do drug discovery programs get off the ground? It depends how much is known about the biological target of interest, but we often use medchem and compchem based approaches to, for example, dock potential molecules of interest into well-defined binding pockets in a protein. With large-scale virtual screening campaigns, this can be done iteratively, and compounds can be assigned a docking score and ranked on that basis. This kind of approach is represented in the top part of this figure in blue. But there's also the potential to use AI to guide the hit generation and hit selection process with ADMET predictions, which facilitate the decision-making process that's associated with narrowing the broad initial group of targets down to a few series of interest. With our AI tool Sapphire, we've made it easy to go from the left side of this graphic to the right and get into the hit-to-lead stage faster with a clear view on aspects like solubility, metabolic stability, permeability, and other key properties as well. A full hit-finding campaign is often built out of a range of different approaches running in parallel, which often include a mix of virtual screening, high-throughput screening, and literature-to-lead-based approaches. Once you've found a hit, it's time to think about setting up a chemistry collaboration, since this is what's going to allow you to advance to candidate selection and beyond. Our chemistry collaborations generally have two primary objectives. The first objective is to accelerate the interrogation of the structure-activity relationships and the structure-property relationships. This is often done by designing syntheses with key diversifiable intermediates or developing diversity-oriented chemical reactions. The second, more development-oriented objective is to develop and execute scalable, phase-appropriate syntheses for the key intermediates and potential candidates. This is important not only for reaching candidate selection, but also for preparing for IND-enabling studies. To learn about what the path to a preclinical candidate looks like in practice, now I'm going to hand it over to my colleague Sujay, who will tell us about a really interesting case study that he led.
Thanks, Matt. Hello and welcome, everyone. My name is Sujay and delighted to be here with you to present a case study from an IDD project that I had the privilege to lead. Over the next 15 to 20 minutes, I'll be walking you through our lead optimization case study, highlighting key strategies, challenges, and breakthroughs that shape the project. I'll be sharing insights, examples, and stories that I hope you will find exciting and valuable. Let's move to the topic. Here is the name of the case study. It's a lead optimization case study, adenosine A2A receptor antagonist for Parkinson disease. Point to be noted here that this project was run by our outstanding team, starting from target rationale, validation, prior search, and early hit finding to clinical candidate nomination. Let's begin by understanding the rationale behind targeting the adenosine A2A receptor in Parkinson disease. Parkinson's is a progressive neurodegenerative disorder that affects millions worldwide, primarily due to the loss of dopaminergic neurons in the substantia nigra. We know levodopa remains the gold standard for managing motor symptoms in Parkinson's disease due to its potent dopaminergic effects. However, prolonged use is associated with significant challenges like dyskinesia and wearing of phenomena. Here's where the A2A receptor comes into play. It located predominantly in the striatum. How it works? A2A receptors are co-localized with dopaminergic receptors in the striatopalatal pathway neurons and it modulates dopaminergic signaling. By antagonizing this receptor, we can potentially restore motor function without directly altering dopamine levels. Use of an A2A antagonist in conjunction with L-DOPA can delay the onset of L-DOPA-induced dyskinesia resulting from chronic activation of the D2A receptor. It can potentially treat depression. How? Indirectly via increasing dopamine D2 receptor activity in the frontal cortex. So we can get multiple benefits from A2A receptor. Now let's move to some important factors and I would like to emphasize here a couple of points. When we started this project, first we analyzed the background and problem. So what was our finding? Number one, at the time, multiple A2A antagonists were already in clinical trials for Parkinson's disease. So competition was quite high. Number two, the chemical space was highly saturated with dense patent coverage, making innovation really challenging. Number three, our goal was ambitious. We are looking for a highly selective compound over other adenosine isoforms like A1, A2B and A3. We are looking for a orally efficacious compound that did not exacerbate dyskinesia, which is a common side effect in PD therapies. So we must need a very strong innovation approaches to address above facts. What innovation approaches we plan? To overcome these hurdles, we adopted a dual-pronged innovation strategy. What are those? Number one, we pursued a fast follow-on approach as multiple compounds are in clinical trials and we are looking for a me too or me better compound quickly. Number two, a knowledge-based design was run in parallel with computational chemistry approaches to identify 2 to 3 diverse scaffolds at the beginning to de-risking potential failures at later stages. Number three, we prioritize synthetic accessibility, developing streamlined route to rapidly explore structure activity relationship. Number four, we are looking for a compound with moderate brain penetration and excellent pharmacodynamics effects. This approach allowed us to move swiftly and smartly through discovery. So what was outcome? The outcome exceeded expectation. We identified a highly potent, selective and efficacious compound with no observed side effects. A preclinical candidate was nominated for Parkinson's disease. Interestingly, another compound from the same program was later repositioned for Immuno-Oncology. That compound is now in phase 1 clinical trials. This further expanding the therapeutic potential of our A2 antagonist platform, which I will discuss as we move. Our work has been published in Journal of Medchem and ACS Medchem later. Also, we have received US and EP granted patent. This slide talks about Medchem approach. To understand where we are headed, it's important to look at competitive landscape. As I said, several promising candidates are in clinics for PD like istadifilin, tozadinan, vipadinan and preladinan. Our approach began by using lipinase-like small molecules, tozadinan and preladinan as starting point, and two series have been started in parallel. We implemented a multi-pronged design strategy that combined knowledge-based insights with computational chemistry to systematically explore novel chemical space and assess intellectual property opportunities. I will discuss series 1 where tozadinan was considered as starting point, followed by series 2 where preladinan was considered as starting point. Let's move to the next slide to see exciting outcome. This slide outlines our approach to hit identification, including hit selection criteria and the screening cascade. We established clear criteria for hit selection. From the outset, our goal was to identify compounds with novel chemotypes to enable strong IP generation, achieve high potency and selectivity. Synthetic tractability is critical. Why? Any challenges in synthesizing the design compound could hinder our ability to rapidly establish structure-activity relationship. We are looking for a compound with acceptable ADME properties, including metabolic stability, solubility, and minimal CYP liability. So what we plan in series 1? Here if you look at the starting point, it has urea linker and compound showed very good binding and functional potency along with very good pharmacokinetic properties with 60% oral viability. Compound also showed efficacy in Parkinson disease model. So what we plan here? If you look at our structure, we deliberately moved beyond urea-based motifs and investigated non-urea derivatives such as amide. This led to the identification of our first hit compound exhibiting strong receptor binding and robust functional potency in early assays. Our design strategy was further validated by molecular modeling. Docking studies confirmed that replacing the urea moiety with aryloxy 2-methylpropanamide was well tolerated within the binding pocket. So what we observe? If you look at the docking pose, the benzothiazole core is buried in the hydrophobic pocket and make pi-pi interaction with phenylalanine 168 and hydrophobic interaction with leucine 249 and isoleucine 274. Also, methoxy carbonyl group form hydrogen bonds with asparagine 253 and tyrosine 271 side chain respectively. Interestingly, Our compound made additional interaction. If you look at the structure of our compound, it has dimethyl group, and this dimethyl group hydrophobically interacted with tyrosine 271 and leucine 267. Please note that the introduction of the 2-methyl and aryloxy groups is crucial for identifying potential pattern space and establishing novelty. Now let's move to the screening cascade. Our team developed a comprehensive screening cascade along with multiple in vivo models. Given our fast follow-on approach, many of these assays were conducted in parallel to rapidly identify promising candidates for lead optimization. Once we synthesize compounds, we screen them first in binding assay and then spot check for in vitro ADME assay. Then potent, selective and stable compounds are tested in functional assays followed by complete profiling in vitro assays. Once compound met initial criteria, a few compounds are taken for the pharmacokinetic studies followed by brain to plasma ratio in rat. When we identified a lead-like compound, it was taken for the defined in vivo studies. Like the PD model was developed and it was haloperidol induced catalepsy model. Then the Parkinson model is the levodopa induced controlateral rotation in 6-OED lesion rats. We have also developed few behavioral model like force swim test, tail suspend test, as well as the chronic effect in combination with the L-dopa on abnormal involuntary movements in 6-OED lesion rat. It's a dyskinesia model. So our target was to at least 2 lead series and take 1 series for the lead optimization. We are also looking for 1 optimized lead and 1 backup compound. Let's move to the next slide for hit to lead optimization. This slide presents our approach to hit to lead optimization and highlights the strength of the novel series under investigation. One of the key advantages of this series is its synthetic tractability. It's requiring only 4 to 5 steps for the synthesis. This enabled rapid library generation, facilitating efficient structure-activity relationship exploration and early lead identification. Once an early hit was identified, we initiated library synthesis to expand chemical space, and we have used ChemSpeed station, and my colleague Matt will discuss more about this instrument in later. In the first and second iteration, we systematically explored substitution on the aryl ring and replacement of aryl with various other aryl and heteroaryl. However, we found phenyl ring is better specifically for the selectivity. Also, we have investigated X is equal to carbon and nitrogen. However, we found X is equal to carbon compounds are more potent and more selective. This effort led to the identification of multiple compounds with single-digit nanomolar potency in both binding and cell-based assay. Potent compounds were tested for in vitro ADME assays. Many of the compounds showed favorable ADME profiles except solubility. Then what we did? Few compounds are taken for the pharmacokinetic studies, and the compound shows very good half-life. However, we observe poor oral viability. So despite this early success, we encounter significant challenges for the poor oral viability, and that could be due to the low solubility. To mitigate these issues, we explored several optimization strategies, and we moved to the third iteration. We identified a promising new analogs by replacing phenyl ring with more rigid linker like cyclohexane. Point to be noted here that magic methyl group is constant. What are the advantages with this new modification? This modification helped to reduce pi-pi stacking interaction due to the presence of phenyl ring. When we replace this phenyl ring with the cyclohexane, we did not see any pi-pi stacking interaction, which can positively impact solubility, and that happened. We'll see the result in subsequent slide. Additionally, we introduce a hydroxyl group and tertiary substitution on the cyclohexane ring to further enhance solubility and improve pharmacokinetics properties, which will be discussed in detail in the next slide. These structural changes yielded a highly promising early lead compound with compelling data. As I said, we explored various substitutions on the cyclohexane ring, and majority of the compound shows high potency in both binding and functional assays, very good selectivity with acceptable ADME properties. Most of the compounds are stable in liver microbiome and hepatocyte. They are highly permeable. Interestingly, most of the compound shows high aqueous solubility with no liability against major CYP and no HARG. From this series, we identified a leak-light compound and you have docked one of these lead-like compound in the A2A binding pocket. When you dock this compound in A2A binding pocket, we observe an additional interaction. The tertiary nitrogen make cationic pi interaction with histidine 264. Now if we look at the table, where we have displayed the data, this compound shows a single digit nanomolar potency in binding assay, picomolar potency in functional assay. They showed a very good fold selectivity over A1, however moderate fold selectivity in A2B and very good selectivity in A3. When this compound again further tested for functional selectivity, they showed excellent functional selectivity over A1 and A2B. Interestingly, this compound has shown multiple improvement in aqueous solubility. Compound was stable in rat hepatocyte, showed high permeability. When we tested this compound in the rat PK, it showed very good oral viability with 61%. This indicating that we successfully addressed the earlier challenges related to the solubility and absorption. Then what next? When this compound tested for blend to plasma ratio in rat, we observed moderate brain penetration. It was around 20 to 23%. And it shows modest efficacy in preclinical Parkinson disease model, which is 6 OSD model. This marked a critical inflection point in the program. As noted earlier, our team had anticipated CNS specific hurdles. That's why we pursued multiple chemical series in parallel, a deliberate strategy to mitigate risk during lead optimizations. On the next slide, I'll walk you through a series 2 where we identified a new scaffold with improved CNS properties and robust efficacy in animal models to continue our lead optimization efforts. When the initial lead series fell short on CNS exposure and efficacy, we swiftly pivoted to another scaffold to address the issues. This pivot led us to novel series with promising pharmacological properties and a differentiated intellectual property position. This new direction not only addressed that limitation of our earlier candidate but also positioned us competitively for future development. After designing this new core, we conducted 4 iterative cycles, each informed by evolving ACR insights. We have synthesized approximately 100 plus compounds with 25 plus compounds per cycle. What do we achieve from that? We identified a set of compounds with high potency and selectivity for the A2A receptor. However, early candidates still face key hurdles. Poor solubility, which is range in between 2 to 6 micromolar, and limited blood-brain penetration, which is improved by another 5% from the previous series. Previous series it was around 20%. This compound showed around 30% blood-brain penetration, which are critical for CNS target. To overcome this limitation, we adopted a library-based ACR approach. This allowed us to systematically explore chemical space while balancing potency, selectivity, and drug-like properties. We explored various options, like on the left-hand side, we have modified the linker. On the right-hand side, we had an opportunity to replace furan with various other heterocycle 5-member or 6-member. We have also explored various substitution on the nitrogen, like methyl, ethyl, and various other substitution on that nitrogen. However, we have observed that magic method is optimum to get selectivity as well as to retain other ADME properties. 2 key modifications proved transformative. 1. Addition of fluorine substitution which enhance metabolic stability, lipophility, and solubility. When we replace the furan with thiazole, we have observed significant improvement in solubility and BBB penetration. In that case, we have found the BBB penetration almost 40%. This changes led to an optimized lead compound and will show exciting data on the next slide. So we successfully identified a compound with compelling data. Compound showed excellent potency in binding assay, showed picomolar potency in functional assay, high receptor selectivity observed in both binding and functional. Compound shows improved solubility and CNS exposure. Compound was stable in both rat hepatocyte as well as in liver microsome. It shows good permeability in Kakoto cell. When we tested this compound rat PK, it shows good half-life and oral viability observed 32%. Most importantly, this compound showed robust efficacy in animal models of Parkinson disease and different behavior model. In haloperidol induced catalepsy model, it shows ED50 4.49 mg per kg. When we tested this compound in 6 OSD lesion rat model, it shows ED50 1.2 mg per kg with no dyskinesia. Compound showed very good efficacy in depression model, both in FST and TST. So what we did next? So later we found a better brain-penetrated compound, and that was nominated as preclinical candidate for Parkinson disease. So what next? Have we stopped here? No, we did not stop here. Despite promising early data, the clinical stage A2 antagonists like pregabalin and tozadinib ultimately failed to demonstrate efficacy in Parkinson's disease, with additional concern around safety and tolerability. Recognizing this limitation, we rapidly shifted focus beyond neurology toward the emerging potential of A2 antagonists in immuno-oncology. This strategic shift was further reinforced by the landmark Heptares AstraZeneca deal in 2015, which highlighted the growing interest and commercial viability of A2 antagonists in immuno-oncology. What is the rationale for A2 antagonists in immuno-oncology? Pharmacological blockade or genetic ablation of A2A resulted in delay of tumor growth and reduction of metastatic dissemination, reversing the immune suppression tumor. Based on our compound's strong in vitro pharmacological, ADME and PK profiles, the emerging landscape, we made a decisive move. We repositioned the same chemotype originally optimized for CNS indication toward immuno-oncology application. Early data suggests that our lead compound retained its potency and selectivity while demonstrating promising activity in tumor models. That compound is now in phase 1 clinical trials, further expanding the therapeutic potential of our A2 antagonist platform. This we achieved within the 8 months after nominating a compound in Parkinson's disease. When we tested this compound in defined safety panel, no issues in drug matrix screening, clean in minims, no cardiovascularity in exploratory dog telemetry, NOL was established for this compound. What was the impactful achievement? Compound represent a major milestone in our program, validating our design strategy and setting the stage for IND enabling. These compounds, along with other adenosine compounds, are procured by US-based company Tarus Therapeutics and followed by another license done by Portage Biotech. So what we achieve? What is the take-home message? From neurodegeneration to immuno-oncology, this journey showcases the power of our strategic innovation, scientific capabilities, and teamwork. Thank you for your patience. Now I'll hand over this to my colleague Matt.
Thanks a lot, Sujay. For me, that's a really impressive and inspiring case study because it shows that it is really possible with the right decisions to produce new, promising preclinical candidates in a short time frame. Returning to the design, make, test, analyze cycle that we saw earlier, I wanted to explore some of the ways that we have to go through these cycles more efficiently, using library-based approaches and scale-up capabilities. Let's take a look at how automation can help us go faster. We use automation in the form of the robotic synthesis platform shown on the right to enable the generation of targeted libraries. The first step in generating a library is the library design. For this, you might already have your own ideas about how to expand on an initial hit with a library of 200 compounds, but you can also talk to one of our experts about what would be the best approach to cover as much useful chemical space as possible with your first library. Further refinement is always possible through subsequent iterations. The execution of the library is then carried out by an efficient reagent handling process and automation of the reactions and workups. A variety of workups and downstream operations are possible in a parallel fashion, including extractions, filtrations, and evaporations. Then, the successful reaction mixtures are purified in our high-throughput purification laboratory using reverse phase HPLC or SFC. Parallel lyophilization then provides the final array of targets, ready for QC and delivery. Using this workflow, it's possible to generate novel, targeted libraries in a matter of weeks, which is really helping you get answers quickly and figuring out which series to prioritize. To help things get going without delays, we have at our disposal over 15,000 building blocks across a wide range of categories, and also a robust inventory of catalysts and reagents to enable the most common reactions of interest for SAR interrogation. This also reduces the overall cycle time, since it means you won't need to wait around for procurement or reagent shipments. Let's check out our high-throughput purification laboratory. Here you can see a very large lab space equipped with semi-preparative HPLCs and SFCs, staffed by a team of purification specialists, who enable the isolation of compounds for the generation of custom-targeted libraries. The lab is organized with efficiency in mind. The compounds are first studied in screening protocols before executing the semi-preparative purifications in parallel. The analytical lab is staffed by a dedicated team that maintains the instruments in top condition and helps to accelerate analytical method development. All of our targeted arrays pass through a final quality control by HPLC before they can be delivered. A fraction of the arrays is also checked by NMR, which we have available up to 400 MHz in open access format. To get a feeling for how this workflow looks in practice, let's take a look at a quick parallel medicinal chemistry case study. In this anonymized example, a client came to us with a synthetic route to a series of interests that they wanted to explore, but it wasn't initially compatible with parallelization. The approach that we took in this case was to design a new route that was optimized for automation, and that sequence is presented on this slide. The scheme involved a series of protecting group manipulations and cross-coupling reactions to produce a final target bearing 3 variable building blocks of interest. In the end, a 70-membered library of novel targets was provided to the client with a 4-week production time, which is pretty remarkable when you think about how long it would take a person to go through this sequence 70 times, treating each individual target as a singleton. Having a partner that can reliably execute on large-scale library requests is very enabling, particularly when you don't need to worry about whether the chemistry is already compatible with parallelization. This really helps you to evaluate different series faster and make better informed decisions. With the number of building blocks that are commercial, many of which we already have in stock, you really have a lot of possibilities. But what if you want to start from a building block which isn't commercial? Figuring out how to make custom, diversifiable building blocks on scale means being able to scale syntheses safely and efficiently. We have experience in scaling reactions and syntheses using continuous stir tank reactors in order to get larger quantities, hundreds of grams for example, of the intermediates that are needed to reach a broad chemical space quickly, either with a library generation approach or with a synthesis FTE team. In this general overview, we can see how the building blocks we choose often have multiple points where variation can be introduced through orthogonal reaction types. We also use our scale-up capabilities to access preclinical API candidates on a larger scale for toxicity, developability, and solid-state studies. For downstream production, we're able to follow through, and we have GMP production capabilities in-house on even larger scale, so that any building block or API syntheses that are established in a preclinical setting can easily be translated into a clinical production environment through an internal tech transfer. Let's have a look inside of our scale-up lab. Here we can see our reactors in action. The reactors are surrounded by a jacket with a thermal fluid whose temperature is controlled by a cryostat. The cryostats can add or remove heat using a secondary chiller to facilitate heat transfer. Key functions like motor velocity are controlled from an external computer, and temperature changes can be programmed and automated. We always study processes on smaller scale before performing them in larger reactors. To that end, we incorporate automated HTE workflows for reaction optimization at an early stage. At the end of a scale-up, we remove the solvent using process scale rotovabs. So until now, we've been talking a lot about small molecule programs and the different technologies and approaches that you can use to advance programs from hit finding to candidate nomination. With that said, one of the most important and exciting frontiers in the field is defined by new modalities in drug discovery. This includes targeted protein degradation, manifested through PROTACs, molecular glues, and beyond, as well as ADCs, and that's where I want to focus now. When we talk about targeted protein degradation, I want to highlight 2 specific approaches that are seeing a boom in the pharma and biotech industry right now, which are PROTACs, proteolysis targeting chimeras, and molecular glues. Both PROTACs and molecular glues are about controlling protein-protein interactions, or PPIs. More specifically, they're able to act as induced proximity degraders. In the case of PROTACs, the key idea is to design a bifunctional molecule that's capable of binding to a protein of interest, or POI, as well as an E3 ligase. The critical role of the E3 ligase here is to attach ubiquitin to the protein of interest and triggers degradation. And here it's also worth noting that tissue selectivity is possible due to differential E3 ligase expression. In the case of a PROTAC, the 2 parts of the drug are tied together by a linker fragment, and rational drug design can generally be applied during the discovery process. In the case of molecular glues, no linker is used, and the discovery process is often much more serendipitous. The reason for that is that molecular glues often rely on much weaker interactions with the protein of interest. For example, they may bind the protein in a shallow allosteric site, as opposed to the active site. To get a feeling for how these drug design concepts can be reduced to practice, Let's first take a look at a brief PROTAC-based case study. The PIM proteins are downstream effectors of oncogenes and are required for tumorigenesis. A number of PIM inhibitors have already made it to the clinic, including AZD1208, shown here. AZD1208, however, like others that have reached the clinic, is a pan-PIM inhibitor, and we wanted to aim instead for a novel and selective PIM3 inhibitor. At the outset of this project, the PIM3 AlphaFold model was used in combination with a published X-ray structure of PIM1 in order to select an exit vector on PIM3 and design a PROTAC series. The initial targets included motifs that could bind to PIM3 as well as cereblon, the protein that forms the E3 ubiquitin ligase complex. We thought that if we could parallelize the synthesis of the PROTACs using this generic approach, we could develop a workflow that would help our clients working in this space to optimize the efficacy and ADME properties of their candidates faster. In practice, the synthesis was adapted to parallelization, which allowed us to generate a targeted array of novel PROTACs. You can probably imagine that projects like this that have significant combinatorial aspects can benefit tremendously from an automated approach. This array was then assayed using our internal biophysics capabilities in order to characterize the ternary complex formation, and through this process we were not only able to generate a series of promising hits, but also provide valuable insights about what changes needed to be made for the next design cycle. In summary, even with complex, challenging compounds, it's possible to use automation to move through an assay cascade and optimize properties faster. And now, zooming out again, I want to turn our attention to molecular glues and see what the workflow looks like in this case. As we mentioned before, molecular glue discovery can be much more serendipitous in nature, and for that, we've developed an ultra-high throughput workflow relying on a spectral shift assay that we execute under 3 screening conditions. In the first condition, we measure the binary binding affinity for the compound with the target of interest. In this short case study I'll show you now, we chose a checkpoint kinase called WEE1. In the second screening condition, we studied the binary binding affinity for the compound with the E3 ligase, which was cereblon in this case. And finally, under the third screening condition, we studied the compound's affinity for ternary complex formation with both the protein of interest and the E3 ligase. Assay cascades using this kind of approach can rapidly identify the most promising compounds for downstream evaluation in other activity-based assays, like our kinase profiler assays, for example. The results of a high-throughput spectral shift screen of molecules for molecular glue behavior allows you to quickly categorize the compounds into families based on whether they exclusively bind to the E3 ligase, the protein of interest, or whether they're able to form the ternary complex. In this Venn diagram here on the right, for example, there were 7 compounds that could form the ternary complex, but did not bind to the protein of interest, WEE1, alone. Nevertheless, these 7 cereblon binders able to form a ternary complex with WEE1 are molecular glues. One of these compounds was profiled even further, and I'll show you a bit about that on the next slide. With the results of the high-throughput spectral shift screen in hand, a program can evolve towards assays that further characterize the ternary complex formation using our biophysics support, as well as towards other assays for protein degradation like Western blots, where in this case we can see that the molecular glue degraded the protein of interest. We also have support for proteomics, which makes it possible to start to understand the fate of the degraded protein, as well as to study any potential off-target behavior. The compound shown on this slide is a close analog of the WEE1 molecular glue from BMS that was disclosed during the last ACS meeting in the first-time disclosure session. And now, with the time we have left, I want to turn our attention to a different modality, antibody-drug conjugates, or ADCs. While in PROTAC drug design, we're trying to localize an E3 ligase to a protein of interest and target it for degradation, in ADC discovery, we're using an antibody to localize a drug to a specific cell type. The virtue of using a monoclonal antibody is that it can very selectively bind to an antigen or epitope on the exterior of a cell. And if that antigen is selectively expressed in a specific cell type, you can effectively deliver a therapy in a targeted way. ADCs leverage different types of payloads, which are generally cytotoxic, and linkers, which are generally attached to the drug prior to bioconjugation. When an ADC reaches a cell with an antigen that can be recognized by the antibody, it can undergo endocytosis, and the drug can subsequently be delivered to the target inside of the cell. Since there are a lot of moving parts here that have to go well for this to work, ADC programs have to have a lot of specific assays in place in order to make sure that each part of this mechanism can be successful, but we'll come to that a little bit later. Now, ADCs are not truly a new modality, since actually they've been being developed for over 2 decades. but recently they're really starting to take off in oncology and beyond. Some targets which have already been validated are seeing increased attention now with new drug-antibody combinations. And there's also a lot of innovation going around surrounding the kinds of payloads that can be delivered and how they can be attached to the antibody. To form an ADC, a drug linker conjugate, or DLC, needs to be bioconjugated to an antibody. This can be done through stochastic conjugation, although this is not an approach that's likely to give a well-controlled drug-to-antibody ratio, or DAR. Molayimids in the linker are often used to achieve the bioconjugation event, so these moieties are often installed at the end of the DLC. Here we're using the sulfur of a cysteine as the nucleophilic residue, but it could also be the nitrogen of a lysine residue, for example. It's also possible, however, to use site-selective rebridging or other site-selective bioconjugation strategies, including engineering mutant antibodies. Here we can see how this allows a narrower DAR, but also multiple payloads per antibody. In the end, the DAR is often closely related to the cytotoxicity of the ADC, so it's important to be able to control it in one way or another. Here we have a general overview of what our workflow looks like for ADC generation. Once we have the drug linker conjugate in hand from our chemistry team, it undergoes bioconjugation in order to form a crude mixture of the ADC, which might have a range of DARs. However, through size exclusion chromatography, or SEC, the ADC can be purified, which also allows the DAR to be narrowed. Once the ADC has been isolated, LC-MS can be used to calculate the DAR. Let's see what this workflow looks like in practice. In this chromatogram on the left, originating from an internal case study, we can see the outcome of the size exclusion chromatography where we were purifying trastuzumab linked to a fluorescent payload. The isolated ADC was then analyzed using flow cytometry as a binding assay. Here we looked at 3 cell lines, one with low HER2 expression, one with moderate, and one with high. And by analyzing the results of this assay, we can see that the ADC that we generated is able to bind to the target cell type. But what do we do now that we know it can bind to the antigen of interest? We want to know that it can get internalized into the cell. For that, we use an internalization assay. In this example, we exposed HER2-positive cells to our ADC and were able to observe internalization as well as cytotoxicity, which we can see in the before and after screenshots. And finally, the DAR analysis was achieved using LC-MS, which in this specific case actually found a DAR of 6 for the ADC. For drug discovery programs involving ADC and degraders, it's really critical to have access to a high containment facility. In our high containment facility, which is co-located with our chemistry labs, we handle ADC projects including payload synthesis, cytotoxin handling, linker attachment to payloads, and DLC isolation. We also support degrader projects with these capabilities through operations like cereblon binding fragment handling, linker introduction, and final degrader synthesis and isolation. And now finally, to wrap up, I want to talk about the scope, impact, and structure of chemistry collaborations at Eurofins Discovery.
Our chemistry collaborations support programs across a wide range of phases, all the way from target validation to IND-enabling studies. We have fee-for-service and full-time equivalent support from end to end, so that you can easily trigger synthesis activities in scenarios with both established and exploratory work packages involved. We've co-located ADME services with our chemistry labs to reduce turnaround time, and we've implemented the robotic system we saw earlier to speed up the early stages. We operate on a range of scales from milligram to kilogram as we transition processes from the medchem labs to the scale-up lab, and we also do process development and route scouting. Afterwards, there's a clear path to GMP manufacturing by internal tech transfer within the Eurofins network. Our global footprint offers all the capabilities needed to run a fully integrated drug discovery campaign from concept to clinic. That could mean running a hit ID campaign employing our 500,000 member high-throughput screening diversity library, executing a large-scale CompChem virtual screening, or advancing a medchem project that might involve knowledge-based approaches, or fragment and DNA-encoded library-based screening approaches, or even production of soluble or transmembrane proteins. No matter the approach, we have the solutions to advance your program. You can access over 300 chemistry FTEs in the EU and India, including our robotics lab for hit-to-lead array synthesis or to solve problematic chemistry, as well as scale-up to help your team accelerate to lead and candidate nomination. Start your program faster by accessing the broadest off-the-shelf offering of robust in vitro assays, and if we don't have it off the shelf, our deep expertise across all target classes and assay development will expedite your program. From a safety talks point of view, well, everyone knows the SERAP panel. Finally, translating your idea into understandable efficacy and a desired phenotype while gauging side effect potential can be done with our experts working in human native cells, co-cultures, 3D models, and disease tissues, as well as generating PK/PD relationships by designing and executing relevant in vivo pharmacokinetic and pharmacodynamic studies.
Thank you very much for watching this webinar, and feel free to stick around and ask any questions you like, or to reach out afterwards and talk about.
Thank you so much, Sujay and Matt, for that excellent presentation. It was very informative for our audience. I just want to remind the audience that you can ask questions through the Q&A box in your screen. And with that, we will now open it up to our first audience member question. I believe this one is for Matt. Given that active protein-protein interaction chemotypes often sit at higher molecular weight log pH than typical drugs, how are library designs and hit triage adjusted to preserve permeability and oral exposure without collapsing potency at the interface?
Hi, yeah, this is a very interesting question. And I think that this, on one hand, comes to one of the major challenges when we come to PROTAC drug discovery. Now, in PROTAC drug discovery, the developability and the bioavailability aspects is certainly one of the canonical challenges that we face. And that has to do with the high molecular weight log P issue. Now, in order to mitigate that, when we talk about library design, we're often thinking about choosing a range of linkers that help to mitigate those effects and give you a range of different properties, so that when we're generating a new library, then we're able to access a wide range of different ADME property sets. And we do that basically by creating a feedback loop between the parallel medicinal chemistry team and the ADME team. On the other hand, there's also the possibility to move beyond the PROTAC design space and into the molecular glue design space. And in that design space, you also have more flexibility towards molecules that are more, let's say, drug-like in the sense of their molecular weight and log P, even though sometimes those discovery programs are sort of predicated on weaker substrate protein interactions. But for that, we're also working on high-throughput spectral shift assays, where it's possible to basically understand very quickly whether or not a large set of compounds has any molecular glue-like properties, which can be used to trigger protein-protein interactions and trigger degradation in cells.
All right. Thank you so much, Matt. I believe this question is for Sujay. This audience member is asking for the A2AR program. Establishing a reliable and translatable preclinical model to demonstrate in vivo efficiency for Parkinson's disease is actually very challenging. What kind of preclinical model do you have to establish this and ensure translatability to human setting?
Yeah, thank you so much. I think it's a very interesting question, especially for when we are working for any neurodegenerative disorder, especially the Parkinson's and Alzheimer's. It's quite complicated, and you can develop many models, especially at Eurofins. We have multiple models available, especially for the clinical. There are the models. The first important factor is the target engagement, which is the receptor occupancy, especially if you look at the A2A, or it's predominantly located in the stratum. So we can do the receptor occupancy, and we can understand what is the concentration at the stratum. You can do the binding assay to understand. That will help for the target engagement. Followed by, there are other various models, so which is the cataleptic model, as well as one of the best models is the 6-OSDA, and followed by you can go for the MPTP model. However, many of these models are, it's not that 100% you can translate it, but you can predict it. Okay, this will work. Even you can later move to the non-human primate. A couple of the models can be tested. Apart from that, there are quite a behavior model which are very easy to establish, as I mentioned in my presentation also, like TST, FST. Even there are some other models also available, and we can easily develop those models. And there are evidence that these models are translating the human, because you might be by this time knowing that many A2 compounds enter into the clinic based on this type of model. I hope I answered your question. Thank you so much.
Okay. Thank you so much, Sujay. I believe this question is for Matt. What is your high throughput multi-reaction screening capabilities? Do you use any machine deep learning method to guide the reaction process optimization activities beyond the standard DOE?
Yes, so that's an interesting question. So when it comes to the high throughput synthesis capabilities, we have two big workhorses. One is our ChemSpeed station, which is used to essentially parallelize sequences that are already able to be, that enable essentially the rapid diversification of building blocks at a late stage to reach a large number of compounds. That's an apparatus that's both used for parallel medicinal chemistry, but also for high-throughput experimentation for reaction optimization. The other major workhorse that we have is a combination of different equipment that are used for other types of parallelization approaches. Now, coming to the question about the AI aspects and how machine learning is used to optimize reactions. This is an aspect that we handle in a couple of different ways. Now, we have some internal software that allows us to do Bayesian optimization of new reaction types, so that we can start with just a few data points and we can essentially build up a statistical model from those data points, and we can start to essentially develop a reaction optimization scheme in this way. We also are working with another external partner that works with text-based, AI-based reaction optimization. So we have a few approaches that go beyond the standard DOE sort of optimization for reactions.
Great, thank you so much, Matt. This audience member is stating, I often see compounds in patents or academic literature that aren't on the market, but which have an interesting biological activity. They're asking, what's the easiest way to get access to those kinds of compounds?
Yeah, so that's also a very popular question that we see from a lot of new clients that are coming in to work with us, because it's true that a lot of the compounds that we want to start a medicinal chemistry program from are known compounds before we reach into exploratory space. And to reach these compounds, normally we operate with a sort of fee-for-service model. And at Eurofins Discovery, we've got a really robust fee-for-service department. Normally, time to quote is 24 hours or less. So if you've got a collection of compounds, you send them over, and basically you'll have a quote within 24 hours, and you'll know how long would it take to have these, and how much would it cost. And basically that means that you could be reading Journal of Medicinal Chemistry in one afternoon, and it could even be that later that evening, you would know how much it would cost to have those compounds in your hands. And within a matter of weeks, you'll be doing your assays.
Great, thank you so much, Matt. This audience member is asking, once you have a compound heading for candidate nomination, what does the path to scale up look like?
Sure. Yeah, so once our clients are essentially nominating their candidates, then they'll start to already be thinking about how am I going to be able to scale up and get my first 100 grams of this compound, whether that's a building block or their actual API candidate. And essentially, we have a scale-up team at Eurofins Discovery that's dedicated to making that transition from medicinal chemistry to process chemistry. And that team is essentially doing a couple things. One is route scouting to make sure that they're finding the best possible intermediates to lead from raw materials to product. And the other is process optimization. So that is the refinement of each transformation leading from each starting material or intermediate to the final product.
Okay, thank you so much, Matt. This audience member is asking, how do you choose when to take a parallelization-based approach to the generation of new compounds as opposed to a more traditional synthetic approach?
Yeah, that's also a question that a lot of clients ask us, and this comes back to basically how diversifiable is the synthetic strategy. If we believe that it's possible to essentially use a diversifiable building block to reach a lot of unique new chemical entities at once, then this is clearly going to be the most efficient approach. Unfortunately, that's not always the case, because sometimes the most interesting space is the most novel space, and that's sometimes the less parallelizable synthetic reaction space. So depending on the types of targets that our clients want to reach, we basically will talk to them and we'll decide, okay, in this case, it's possible to use a parallelization approach, and that's where we try to leverage automation and high throughput purification techniques. Or do we need to take a more manual based approach that leverages more the experience and expertise that we have rather than the automation?
Okay, thank you so much. That's about all the time that we have. Thank you again, Dr. Sujay Basu and Dr. Matthew Horowitz for that wonderful presentation. thank you all the participants for being a great audience. You asked some excellent questions. And also a thank you to On24 for the technology and production services. I just want to let you know that if we didn't get to your question during our Q&A session, that it will be answered by email. A big thank you to Eurofins Discovery for the sponsorship that made this interactive webcast possible. Be sure to check CNN or CNN Online for the information on the next edition of CNN webinars. And for CNN webinars, I'm Melissa O'Mara. Goodbye.